Take it away, Pam.
Okay, well, I've got a couple of caveats here. One is that Schwanmin sent out a request to faculty, about a month or 2 months ago, I don't know, a long time ago. And I said, well, I'm going to be getting a talk ready for a meeting, and so if you really need me, but I keep getting up here, and he assured me that he didn't need me. And then 2 days ago, I saw my name on the list, and I hadn't been working on the talk because the talk is for a meeting in a couple of months. However, I've been working with this group in Mallorca, and it's with the extensions in Italy, and the manuscript that we're working on is only 70 pages long, and I must confess that I took a lot of this talk from Luis Pomar's talks that he's been giving this summer, and put a little bit of my more biological twist to it. But Luis is absolutely fanatical about animation. And so there are times when, and very complex slides, and I probably didn't eliminate enough of them, but so this is with this caveat. If you want to go, I won't feel offended. Okay, let's see where we should go here. Is this what? Yeah, there we go. Whoops, that wasn't what I wanted. Okay, well, anyway, what I'm going to talk about is the last. Actually, I'm going to talk more about even the last couple of 100 million years, but focusing on the last 65 or so million years, and what chemistry, climate, and biology has to do with anything. And some of you know that, and this is obviously my slide with a little bit of Luis's figures thrown in here. Mixotrophs are organisms that both feed and photosynthesize, and I have long contended that mixotrophy is a fundamental adaptation to living in low nutrient environments where the only concentrations of nutrients are in particulate organic matter, or in organic matter anyway. And so if, like a Venus flytrap living in a plant living in a very low nutrient environment, if they can capture a particle of organic carbon, they can photosynthesize to produce the day-to-day organic material for respiration, but to grow they need to capture some nutrients. And in very clear low nutrient environments, the best concentrations of nutrients, although they're sparse, are plankton or pieces of particulate carbon. And at the same time, I've been working on this issue for, you don't want to know how long, but the microbiologists have found out a lot of stuff since I started working on this. Things like we're all holobionts. A holobiont is an organism with all its symbionts, with all its little things that live with us. And is it what, Maya, is it that there's 100 times or 1,000 times more microbial cells in our body than human cells?
Yeah, 100.
100 times, 100 times, okay. And so, when I first started, it was all about the coral or the foram and their algal symbionts. But now it's about the whole host of symbionts that they might have. We know zero about the microbial symbionts in forams, at least in the algal symbiont bearing forams. There's a little bit done on some of the hypoxic taxa. Okay, then whereas a fair amount of work has been done now with the microbial associations in corals. But essentially, we've known for, well, as long as I've been a scientist, which is a really long time, as I mentioned, that corals, for example, get about, their zooxanthellae produce probably twice as much organic matter from photosynthesis, at least in shallow water, than the corals themselves can use, the corals and the algae can use. That is excreted as mucus. And I have long suspected that the bacteria would feed on that mucus and then attract other things to feed the corals, or maybe even the corals were feeding on the bacteria. You see, bacteria, because they're a very minimal size, can pull out, as long as they have a sugar source, they can pull what little nitrogen and phosphorus there is out of the water at much lower concentrations than, say, a full-blown algal cell. So these guys are the kind of the ultimate solar generators, but they can produce organic matter. They can produce oxygen, essentially, and you'll see coral reefs are among the most productive ecosystems on Earth. And back in the 80s, people were going, like, we can fertilize them and grow more fish. Except when you fertilize a reef, what do you get? Algae. Yeah. Okay, because very high gross primary productivity, very low net primary productivity. In fact, essentially zero. Okay, so, but they can feed, they photosynthesize, and they can farm bacteria on their excess photosynthate. We've also known for a long time that forams, if they're just sitting still and not moving around catching things, they will produce an organic sheath around them that, or at least some species do, that then they farm bacteria in that organic sheath. Okay, or they probably farm bacteria. There hasn't been any work done on the actual bacteria. Okay, these critters also, especially over the last 50 million years or so, have been the major producers of carbonate sediments. Whether you're looking at the good thing about deserts, or desert kind of environments, is that the geology is not all cluttered up with plants. And so this is this is actually what we call a ramp, a carbonate ramp. The West Florida shelf is a carbonate ramp, but the accretion of the limestones on that carbonate ramp is pretty much pure foraminiferal, larger forams. And of course, I just threw in a couple of I missed getting the IMARs. I think it's underneath something somewhere. These are images of reef buildups from space from the Frank Muller Carter's group. Okay, so this is my only mention of deeper geologic time. We've seen a change in what we call the carbonate factories through the Permian, Triassic, Jurassic, Cretaceous, and what I'm talking about here is primarily this last bit of time. The LBF means larger benthic forams. C is for coral, RA for red algae, coral and red algae, and again C for corals. There's some interesting things about corals in this whole story, and that is that we see the first evidence of the symbiosis way back here in the Triassic, and there's some radiation and reorganization and so forth, and but the corals never really seem to be very efficient producers of limestone, actually, until fairly recently. And so we're going to look again at this latest time, just some examples of modern larger forams and coral and algae. And now we're going to look at some of the paradoxes. Again, I've been playing with paradoxes of carbonates and symbiosis for all those many years that I've been, since I was a graduate student. And one of the things that was pointed out, one of the things that was pointed out by a geologist named Stan Frost, a carbonate sedimentologist, way back in the 70s and early 80s, he pointed out that the modern coral reefs really didn't start ramping up until the high latitudes started getting cold. So here you've got a tropical biota that depends on on polar cooling. What's going on here? Oh, and the other thing, the diversity image, he pointed out, and other people have pointed out, that there's almost no relationship between diversity and carbonate production. In fact, there are places where you have essentially one or two species of coral that can accrete limestone just about as fast as a high diversity. In fact, often faster because of some other aspects that I can't go in, that I don't have time to go into. But then the other thing that's interesting, and this is Luis Pomar and I've been throwing these ideas around since about 10 years now. And that is that the larger benthic forams kind of peaked here in the Paleogene with a secondary peak in the Neogene in terms of diversity and also in terms of apparent production or a production of carbonates, although this figure is a bit misleading because there are plenty of forams out there on the reefs, especially in the Indo-Pacific. And somebody one time made the statement, they were talking about the really wonderful molluscan fossils, the shells, the snail shells and so forth from the Dominican Republic. And I asked them if they ever saw forams in the sediment that they were getting the shells out of. And the person just looked at me and said, well, if all the larger forams from the Dominican Republic suddenly dissolved, the Dominican Republic would disappear from the face of the earth, which was basically or sink below the waves. That's what they said. I'm sorry, not disappear from the face of the earth, sink below the waves. But there seems to be something of an inverse correlation here. Oops, I'm still hitting the wrong. Okay, so coral reefs, though, thrive in an icehouse world. So the big question is why. And it's probably because algal symbioses have costs as well as benefits. And a scientist at AIMS, at Australian Institute of Marine Science, working on coral bleaching, has proposed that coral bleaching is a destructive endpoint of cellular processes that are essential to the establishment of an algal symbiosis. And if anybody wants to go further on that, I don't have time to talk about it here, but I can certainly give you that paper. But it has to do with the fact that corals have a kind of an inefficient Rubisco, which is an enzyme really critical in the production of organic matter, glucose, from the capture of energy by photosystem 2. And basically he contends that because of this poor ability, that if the the carbon concentrating mechanisms of the coral are unable to be delivering, and of the cell walls are unable to deliver enough CO2 to for all the light energy that's being captured, then the Rubisco will break up oxygen, and the oxygen radicals will go berserk and cause the bleaching. So that's all I'm going to say about that for the moment. We have a diverse array of kinds of carbonate buildups. People have recognized these different things for many years, and the things called reef mounds, or what we're calling cluster reefs here, are often in deeper water. They can be aphotic, so non-zooxanthellate corals, or deeper water, so-called mesophotic reefs. Frame reefs also. This is wrong. This should be segment reefs. I need to get to, and then the true framework reefs of our modern world. Okay, so the other thing that's really critical is light. Light is like that's how we get the photosynthesis, and that's what drives both the symbiont-bearing organisms, the forams and the larger forams and coral, but also the calcareous green algae and the coral and reds. And Luis Pomar actually has coined these terms of rhodocline, the oligophotic zone, the chlorocline, euphotic zone, and really this kind of in-between zone is, or the upper part of the oligophotic zone, is the what people now call the mesophotic zone. The coral researchers of mesophotic coral reefs are a big thing right now. Okay, there's also the whole issue of what our paleoproxies can tell us about carbonate production, and so we're working on incorporating, the oxygen and the cooling is easy, because as we see the cooling, we see the increase in corals. The carbon is still somewhat problematic. I think this is more important than Luis does, but this drop in calcium versus the increase in magnesium, and of course the PCO2 proxies are kind of all over the place, and so I haven't quite wrapped my head around those to the degree that Luis thinks he has, but I don't completely agree with him. Anyway, so we're going to look at the reefs through time and when we have significant coral buildups, particularly significant coral buildups in the Mediterranean, because this is based largely on the people that I'm going to recognize at the end. All are working on and have worked for many years in what I'll call the middle tethys, or what we can call the middle tethys. In fact, I think. Okay, so at least in the Mediterranean region, virtually all of the outcrops that they have explored that have corals in them primarily show evidence of being the cluster reef types and of lower light intensities. And whereas the framework reefs are more characteristic of the late Neogene and the Pleistocene and Holocene. So now we're going to take a quick tour, and I'm not going to go through all the details here because there's way more than I could, but this tour of the central Tethyan region here and some of the carbonate buildups there. And this is kind of probably some of the newest thing is really recognizing that there was some recovery of coralgal reefs of Cretaceous affinity in the Upper Danian, but not true framework reefs. And I've just hit double there. Okay, then in the Upper Paleocene, there was some recovery of reefal ecosystems, and that was followed actually by the development of really algal foram systems. And let's see. So here we see the heyday of the larger forams right here in the Eocene, and what we see is that they went from in the late Paleocene, there were more coralgal reefs with the larger forams were more tropical. Then as there was overall warming, the carbonate production was dominated by larger forams, and the coralgal reefs were pushed into kind of middle high latitudes. And ultimately, at the Paleocene-Eocene thermal maximum, we see actually the larger benthic forams. Some people say that there was a extinction. Other people say they really expanded, and so there's some combination, and that still is a work in progress. As we go into the well, this is more the corals of the early Eocene. There were some patch reefs, and this is an example from the Southern Pyrenees, if I recall correctly. Unfortunately, I haven't gotten to go to all of these places that I'm going to show you pictures of. Some of them, but not all. And then the Letetian, were pretty much dominated by the larger forams, but with some coral-bearing mud mounds. And